Hey guys, welcome to Tricky Topics for Chapter 18. So for Chapter 18, we're going to be going through mutations as well as mismatch repair. So this video is going to be going through all our different mutations, and then another video will be going through mismatch repair. So let's start looking at mutations. And there are three different types of mutations, and we're going to go into each of these in more depth. The first one is mutations in the DNA. The second is mutations in your amino acid. And your last one is going to be phenotypic mutations. So you can see it kind of builds a little bit from DNA mutations to now we have an issue in our amino acid to now there's some sort of phenotype mutation. And the phenotype mutation it doesn't just have to refer to what you look like, it could be refer to what the protein looks like and things like that too, the protein's individual function. So let's get started with DNA mutations. There's two main categories: substitutions and frameshift mutations. We're going to start with substitutions. So in substitutions, you're changing out one nucleotide for another. So you're not going to be extending the length of the DNA. You're not going to be shortening the length of the DNA. It's the same length. You're just changing one nucleotide out for another. And there's a couple different ways this can happen. It could either be a transition mutation or a transversion mutation. And a transition mutation, you're going between the same type of base. So for example, if you have a purine and you change it to another purine, that's a transition. You're within that same type of base. With the pyrimidine, if you were to change it to another pyrimidine, that would again be transition because you're in the same type of base. So remember to know your purines and pyrimidines. For purines, it's A and G because angels and gods are pure. And for your pyrimidines, it's T, C, and U because TCU is not pure. So those are your transitions from one to the same type. Transversion is going to be going to the other type of base, switching to the other version. So the way that I remember transversion is that it switches from one version to the other, and transition just must be the other option. So transversion would be if you're going from purine to pyrimidine or pyrimidine to purine, and you can see the examples of that there. So that's a substitution. Now let's look at frameshift mutations, and there's two different types. And with frameshift mutations, this is going to shift the entire sequence over. So if I were to insert one nucleotide, everything needs to move down one. If I were to remove one nucleotide, everything is going to shift over this way. And where this is going to have a really important effect is going to be within your codons, because I know that my codons are read in groups of 3. So if I were to suddenly remove the third nucleotide of something, what was the first nucleotide in the next codon is now the third nucleotide of the one right before it. So everything shifts over one spot, and that means that your codons are going to look different, which means that your amino acid sequence is going to look different. And we're going to talk about those in a second. So with frameshift mutations, you can have an insertion, which is adding a nucleotide, or deletion, which is removing a nucleotide. So now let's talk about amino acid mutations. Your first one is going to be if you have one amino acid and you change it out for just some other amino acid. For example, going from proline to alanine. You're still coding for an amino acid, but it's a different one. And that's called a missense mutation. Your next one is if your codon is mutated, but instead of coding for another amino acid, it's now a stop codon, meaning that your sequence is terminated and your polypeptide chain is going to be released prematurely. We have a stop codon where we originally had something like an arginine. And having a stop codon early, it's nonsense. We don't want that, so it's a nonsense mutation. The third type of mutation is a silent mutation because your codon is mutated, but it still codes for the same amino acid. So if you remember your codon chart, we called it degenerate because you can have a lot of different codons code for the same amino acid. So sometimes you can mutate a codon and have a different nucleotide and it still code for the same amino acid. So you still had a mutation, it just doesn't have an impact at the amino acid level. But to represent that a mutation still existed, we call it a silent mutation to say, hey, there was a mutation, but it coded for the same thing in the end. So for example, we had histidine, we changed the codon, we still have histidine. So now we're going to talk about phenotype mutations. The first one is a gain of function, meaning your protein can do its job better or it can do something new. The next one is loss of function, losing part or all of its function. Your next one is lethal, causing premature death. And the next one can be a little confusing. It's called a neutral mutation. So here we have a change in the amino acid, but because our new amino acid is very similar in its properties to the original amino acid, we don't really see a change in function. For example, if you have a positively charged amino acid originally, and we changed it to another positively charged amino acid, because they're very similar in that they both have positive charges, we wouldn't think that the function of the protein would be changed that much. So keep in mind the difference between neutral and silent. With a silent mutation, you're coding for the same amino acid. With a neutral mutation, you're coding for a different amino acid that has same or very similar properties. And then the last 3 types of phenotypic mutations we're going to go over in the next slide: forward, reverse, and suppressor. So starting with the forward mutation, this is going from a normal phenotype to a mutant phenotype. And reverse is going from the mutant phenotype to the normal phenotype, and suppressor is going from the mutant phenotype to the normal phenotype. So you're probably looking at those last two and saying, those say the same exact thing, how do I tell them apart? So both of them are going to be referring to some second mutation that brings it back to normal. The first mutation would be your forward mutation, where we change it to a mutant. So first mutation is going forward, the second mutation is bringing it back to normal, and we could do it through reverse or suppressor. So with a reverse mutation, we're going to restore your phenotype, which we already know we're going back to normal phenotype for both these, and the genotype. But for suppressor mutations, you're going to restore the phenotype, but not genotype. I think this makes the most sense with looking at an example. So we're going to look at these codons. Let's say we started with AGC, which codes for phenotype A, and we change the A out for G, and those are both purines, so that would be a transition mutation. And now we have phenotype B. A reverse mutation would be changing that G in the front back out for an A, in which case we have phenotype A again. So this first mutation where we went from normal phenotype A to mutant phenotype B, that's a forward mutation. And the second one, we have a reverse mutation because we have the same genotype at the beginning and the end, as well as the same phenotype. So because genotype and phenotype are restored, it's reverse. Let's compare this to suppressor. We have the same beginning where we have AGC and phenotype A, changing to GGC with phenotype B, which is a forward mutation. The difference is in this next part. We change it to GGG. So we're changing the C at the end to a G, and C is a pyrimidine and G is a purine, so that would be a transversion mutation. And notice we're back at phenotype A, so we did restore the phenotype, but your genotype at the beginning was AGC, and now we're GGG. So we did not restore genotype. We had AGC to GGG. That's a different genotype, although we still have the same, or we went back to the normal phenotype. So in this case, that first mutation would still be a forward mutation, but the second one is suppressor because genotype did not also go back to the normal. The genotype is still mutant. So specifically, in this example that I gave you right here, this is a type of intragenic suppressor mutation because it's happening in that same codon. And there are actually 2 types. There could be intragenic or intergenic suppressor mutations. For intergenic suppressor mutations, that means that second mutation is happening at a different location. For example, if the forward mutation happened at allele A, and then your suppressor mutation happens at allele B, 2 separate locations, we call that intergenic, 2 different genes. And intragenic happens at that same location within the same allele, at the same codon, in the same gene. And this is the example that was just on the previous slide, where we're looking in that same codon where we have the reverse and the suppressor mutation happening. All right, and the next video we'll be going through mismatch repair.
I hope you found this video really helpful. The concepts and information presented in these videos will be true no matter what genetics class you are taking. However, the concepts presented in this video are referencing material currently covered in Baylor University's coursework. Remember, if you are currently enrolled as a Baylor student, we offer free tutoring services. Our tutoring center is located on the first floor of the Sid Richardson building. You will find all the details you need to know about these services on our website, www.baylor.edu/tutoring. You may schedule a free 30-minute one-on-one tutoring session online through Navigate, or just drop in during our open business hours. For more information about our current services, please visit our website. Thank you.